This conference will now be recorded.
Hello students, we are moving to another chapter of botany, chapter number 11, transport and plants. So here the topics which we are covering, that is for NEET syllabus, we are covering the means of transport, plant water relations, then transpiration, guttation, all these. Okay, so from this like from this chapter, the type of question for NEET 2023, some questions came from this chapter, and 3 questions from topic 1, that means the means of transport. And in part 5, you will get, like, you're going to write the NEET exam like 2-3 sessions. Types of questions will be different. So different types of questions, easy questions will be there, little bit you have to think about the concept, then assertion type questions will be there. So all these type of questions you will get in your NEET exam. So here in 2023 NEET question paper, questions were visible from this chapter, and questions from first topic, that means the means of transport, that topic, and 2-3 questions from each topic, like plant water relations, transpiration, guttation. From all these topics you uh, there were questions. Okay, long distance transport of water, all those things you are learning. Then phloem transport, transpiration. From all these you got 2-3 questions in the previous years. So here you know the importance of each question and mark, right? If you are getting 2-3 questions, means how many marks per question? You will get 4 marks, right? So better such, this is a easy chapter, you can go for a easy revision, and exactly you can say like a quick review you can do for this, no need to spend so much time, right? So such chapters, and in previous year question paper you are getting 2-3 questions, means definitely you'll get questions in this year also, right? So what you need to do for such chapters, just plan the hours and go for a fast review, a quick review with the previous year question papers. Okay, so you'll get an idea. Like, no need to sit and read for 2-3 days and all. Just go through the chapter once and go through the previous year question paper. Okay, so let's start with the chapter. First, we will learn about the means of transport. Here, means of transport. What is the transport in plants? What do you mean by the transport in plants? Mainly the transport in plants that involves the movement of water, minerals. In plants we are saying. Okay, so in plants the movement or the means of transport. Transport means what and all will get transported. Water transport will be there. Then minerals and organic solutes. Right? So these many things are getting transported to different plant parts, right? So how it is getting transported? This transport is through vascular system, right? Vascular system is transporting all these things to different parts of the plant body. Vascular system, what is this? Xylem and phloem. Xylem and phloem. So transport in plants mainly involves the movement of minerals, waters, organic solutes, all these things to different parts of plant via the vascular system. Okay, vascular system involves xylem and phloem. And within a plant, transport occurs at different level, right? That may be a short distance transport, that means within a cell or within different cells of a tissue. Okay, so distance, that also matters. That may be a short distance transport may be short distance transport, or it may be a long distance transport. Okay, so what do you mean by a short distance transport? Within the cell or tissues. Okay, it may be within the cells or the cells of a tissue. Okay, or what is the within the cell or within the different cells of a tissue, that is called a short distance. Long distance means within different tissues. Within different tissues. Within different tissues means it may be within the same organ or to the other one. In same organ or other one. Okay. And short distance transport, we said this short distance transport may occur by active transport and passive transport. Okay, short distance transport may occur. This may be by active transport or passive transport. Active transport or passive transport. Okay, here passive transport, that you can say simple diffusion, facilitated diffusion. Also, we'll learn about that. First, let's see what is active transport. Okay. See here in this chapter, you can see what is active transport, what is passive transport. So all these things you can learn. But here, no need to get confused. Write the points clearly. If you are making short notes, make the points with clarity. There should not be any confusing points in your notes, so that exam time you won't get tense. Why? With one easy reading or within 1 hour you can learn all the concepts of this chapter, then you can go for solving the previous year question paper. Okay, so how you need to learn? Here some transport methods we are saying, like active transport. Suppose we are saying about active transport, that is a process of transport which requires energy. So what you will write? Active transport requires energy, right? And that allows the transport against a concentration gradient. Against a concentration gradient. Which one? Active. That means it's actively, just try to remember, active transport. It's an active way of transport. Okay. So usually how transport will occur, that means how, see, if from a higher place water will flow to a lower place, right? So there is no need of energy, it will move because water can't stand there, it is moving down, right? So that is, there is no involvement, there is no requirement of energy. But in an active transport it's not like that. It is against the concentration gradient, and from a concentrated region, the in a solution it will move to the low concentration regions, right? That's why that's how the concentrations are becoming equal in a, if you are keeping two solutions, if you're mixing two solutions like that, you have learned, right? So in the same way, here in active transport we are writing that it is against the concentration gradient. Against means it's not in that way. Okay, in opposite way also it can happen. So here it allows the transport against the concentration gradient. That means from lower concentration to higher concentration. From low concentration to high concentration area. Okay, so that is happening. Okay, and this process is facilitated by highly specific carrier proteins. It's by carrier proteins. Okay, so something is actively involved in this. Okay, that is facilitating this kind of transport, which is called as active transport. So here the process is facilitated by highly specific carrier proteins. Here carrier proteins are specific. Okay, all carrier proteins won't work for all the active transport. It is specific. Okay, so in that way you need to learn. Then comes to this is the active transport. Next we are moving to passive transport. Second type transport is passive transport. So what is this passive transport? See, passive transport is a process that does not require the involvement of energy. No energy requirement. Okay, energy is not required. The passive transport is a process in which no energy requirement is involved. Okay, and that mainly involves downhill transport. Downhill transport. Okay, downhill. Just now I said downhill transport means from a region of higher concentration to a region of lower concentration. Region of higher concentration to lower concentration. That is the downhill transport. Okay, and here examples we can say example for passive transport, that is diffusion, then facilitated diffusion, then osmosis, plasmolysis, imbibition. Okay, so here diffusion, facilitated diffusion, osmosis, plasmolysis, imbibition. So everywhere in all these, which is involved, this is coming under passive transport. Okay, so there is no energy requirement, and the process of all these, that is the downhill transport, region of higher concentration to lower region, lower concentrated region. That's how the movement will take place, right? So first let's see what is diffusion. Here under passive transport you need to learn diffusion, facilitated diffusion, osmosis, plasmolysis, imbibition. Okay, so this basic concept is same. It's a passive transport method without energy requirement, and that is the downhill transport method. Clear? So let's see first diffusion. Okay, diffusion means the process of movement of molecules from the region of higher concentration. I said that is passive transport method. So diffusion means from high concentrated region to the region of low concentration, to low concentration region of molecule. See, molecules will move from the region of their higher concentration to the region of their lower concentration, and that movement depends upon concentration. First, in one place, higher concentration should be there, then another place, lower concentration should be there, and based on concentration, kinetic energy of molecules, temperature, and pressure. Okay, there are criteria. So, first one is concentration. This diffusion is based on concentration, concentration then kinetic energy, then molecules, kinetic energy of molecules, then temperature and pressure. So based on these four factors, diffusion is happening. So this is a very important question, previous year question. Okay. So here you will get diffusion from the below mentioned ones. Here one another option will be there. These are the basic things, and based on these, diffusion is happening. So concentration is must, because diffusion is happening from high concentration to a low concentration region, right? And here kinetic energy of molecules, that is also important. Temperature, that is important. Pressure is important. So based on these four factors, diffusion is happening. So this is the previous year question, and from the option you need to select. You read the question, which is the factor which is not affecting diffusion, or from among the following, select the factors which are affecting diffusion. Sometimes they'll give this A, B, C here. Instead of this, something else will be there. Okay, some unknown option will be there. So A, B, D. D is the wrong answer. D is some other factor they have given instead of pressure. Okay, so A, B, D, some wrong combination they'll give. So first you read which is affecting or not affecting the diffusion. Okay, based on that you have to choose the answer. So while writing this kind of question, you should be very careful, read the question properly, then go to the answers. Okay, so that's how diffusion we can define: the process of movement of molecules from a region of their higher concentration to the region of their lower concentration, and that depends upon kinetic energy of molecule, temperature, pressure, and the concentration. Clear? And under diffusion we need to learn diffusion pressure. Diffusion pressure. Diffusion pressure, dp. Okay, so in short form we can write as dp, diffusion pressure. That is a pressure exerted by a substance. Pressure exerted by substance due to the tendency of the particle to diffuse. Okay, so that is a pressure which is exerted by a substance due to the tendency of that particles. That substance will be having the particles, so that particle need to diffuse, so they will exert a pressure. That is called as diffusion pressure. Okay, and this diffusion pressure, that is directly proportional to the concentration, then the temperature. Here, this is another question: what is diffusion pressure? Okay, the pressure exerted by the substance due to the tendency of its particle is called as, or the tendency of the particle to diffuse, that is called as, options you will get. So among that options, the right option will be the diffusion pressure. Okay, that is one question, previous year question. And another one, the factors, or the factors which are given below, they are directly proportional to the concentration temperature. So you have to pick the factors. Which are they? Number of diffusion particles, temperature, concentration, all this. Concentration, then temperature, then number of diffusion particles. All these factors are directly proportional to what? These are directly proportional to concentration, temperature, diffusion pressure. Directly proportional to diffusion pressure, or dp. Okay, what and all? Concentration, then temperature, number of diffusion particles. All these are directly proportional to diffusion pressure. Okay, if concentration of that solution or that particular thing is more, diffusion pressure will be more, right? Temperature is increasing also, pressure, diffusion pressure will be more. And if number of particles are more, means we said, what is the definition? Pressure exerted by the substances, the particles of that substances will exert one pressure, so that pressure is more, means definitely diffusion pressure will be more. So all these are directly proportional to diffusion pressure. Okay. So this is diffusion pressure. So we have learned about diffusion. Then next term, what we have said, that is facilitated diffusion, right? Second term under passive transport, that is facilitated diffusion. So what is this facilitated diffusion? This is also the process of diffusion of molecules from the region of their higher concentration to the region of lower concentration. Okay, so that is same here, facilitated diffusion. This is also a diffusion process. Diffusion from high concentration to lower concentration. Okay, but here the thing is, this diffusion process, here what we said, it is facilitated diffusion. This is also common diffusion process, but it is facilitated with the help of something. Okay, something is helping. Okay, so to happen this diffusion, something is helping or something is facilitating this diffusion process. So what is that? What is the thing which is helping for this diffusion process? So various transport proteins, here we can say transport proteins. Transport proteins are involved. They are facilitating this diffusion. Okay, so various transport proteins are involved in this process, and which are they? Let's see. Carrier proteins. Carrier proteins. So carrier proteins, these are membrane proteins. So carrier proteins, then channel proteins are there. Channel proteins. So all these carrier proteins and channel proteins, they are the proteins which are helping for this facilitated diffusion, or these are the helping things which is helping to this facilitation of the diffusion. That's why we are calling as facilitated diffusion. This diffusion is facilitated by these kind of proteins which are called as carrier protein and channel protein. So let's see first the carrier proteins. Carrier proteins are membrane proteins. Membrane proteins. Okay, carrier proteins are membrane proteins, and that are bind to a substance and they will transfer it to the other side of the membrane. Bind to these proteins that bind to the substance, and after binding, what they will do, they will transfer that substance to the other side of the membrane. Transfer the substance to other side of membrane. Okay, see here the protein is there, so it is binding to the substance, and what it will do, other membrane is here and it is moving this to the other membrane. See, from here it is taking to the other side of the membrane. So this may involve uniport system. This carrier protein that may involve uniport uniport system, then symport system, or antiport system? Uniport, symport, antiport system. Okay, so carrier protein, we said the membrane proteins, they will bind to the substance and they will transfer it to the other side of the membrane. So here this carrier protein, they may involve a uniport system. We said, what is a uniport system? Diffusion of one molecule. Diffusion of one molecule. Okay, uni means one, single one. So diffusion of one molecule, that is called as uniport, right? Then the symport system, we said. Symport means diffusion of two molecules in single direction. Two molecules in single direction. That is symport. And antiport means diffusion of two molecules in opposite direction. Diffusion of two molecules in opposite direction. That is the antiport system. Okay, so carrier proteins will bind to the substance and they will transfer the or transport the substance to the other side of the membrane. So that involves uniport system means diffusion of one molecule. Symport system means diffusion of two molecule but in same direction. And antiport, anti means again. So both are going to both the molecules. Diffusion of two molecules, that means diffusion of two molecules are taking place in opposite direction. In which one? In antiport system. Okay, so this is the carrier protein. In facilitated transport, carrier proteins are facilitating the transport. Next, the channel proteins are also helping to facilitate the diffusion. Okay, so that we will learn. What is that? In facilitated diffusion, second type of facilitated diffusion, that is channel proteins. Channel proteins are also helping to facilitate facilitated diffusion. Okay, so these are also membrane proteins and that act as a pore. Okay, this is also membrane protein. Which one? Channel proteins. Okay, so these proteins will act as a pore through which that molecule can travel. So in carrier protein, what it is doing? That protein molecule, suppose this is the protein molecule A. This is binding with the substance which has to be transported. So this protein molecule is binding with the substance. This is a substance. Okay, then these together, it's moving to the other side. But here in the channel protein is also present in the membrane. So what it is doing, this channel protein is making, or this is acting as a pore. Okay, this channel protein is making a channel or a pore through which that particle or the molecule can travel. So the substance here, the substance can move through this channel which is made by that channel protein. Okay, so that includes porins. The porins are present where it is present in the bacterial membrane. Bacterial membrane. Porins are present in the bacterial membrane. Then aquaporins. Aquaporins, these are water channels. This will work as water channels. Okay. So diffusion, we said here passive transport. In that we are saying about diffusion, facilitated diffusion. In facilitated diffusion, we are learning about the things which are facilitating that diffusion. And here, which are the things which is helping or facilitating the diffusion? Carrier proteins and channel proteins. Okay. And diffusion helps in gaseous exchange. Okay, it helps in gaseous exchange. Then transpiration, translocation of food material, transpiration. For that also diffusion is helping. Then the transportation. We said about the facilitated transport and all, right? Transport of material. All these are done with the help of diffusion. So diffusion helps in the gaseous exchange, translocation of food material, then transpiration. Okay. So in passive transport, about diffusion we have learned, facilitated diffusion we have learned. Next we need to learn about osmosis. This is also coming under passive transport. Osmosis means this is a passage of solvent molecule from the region of higher concentration to a region of lower concentration. Solvent molecule from high concentration to low concentration region. That is called as osmosis. And this passage that is happening through a semi-permeable membrane. Through semi-permeable membrane. Okay, this passage is happening through the semi-permeable membrane. So osmosis means the solvent molecule is moving from the region of higher concentration to the region of lower concentration through semi-permeable membrane. That is called as what? Osmosis. Okay, so here passage of solvent molecule from the region of their higher concentration to the region of lower concentration through a semi-permeable membrane, that is called as osmosis. This is one important question, previous year question. And we said about the carrier proteins and channel proteins. In that uniport, symport and antiport system, that is also important questions, previous year, coming under previous year questions. Okay. And about porins, we said that porins, they are also very important question, which is present in the bacterial membrane. Okay, porins and aquaporins. So here, when we are learning about osmosis, that is the passage of solvent molecule, not the solute, the solvent molecule from the region of the higher concentration to the region of lower concentration, and this passage is happening through a semi-permeable membrane. Okay, and here the movement, how the movement of this water molecule or the solvent movement is happening. Solvent, here we can say the water. So that solvent movement or the movement of water between two systems that occurs through difference in the water potential. Difference in water potential, that makes the movement. Then the movement of water across a plant cell, that also depends on osmotic concentration. What do you mean by osmotic concentration? Osmotic concentration means amount of dissolved solute per unit volume of that solution. That is the osmotic concentration. That solute which is dissolved per unit volume of solution. That makes the concentration, osmotic concentration. So based on the osmotic concentration, we can divide the solutions into three types. Okay, so based on osmotic concentration, solutions are of three types. Which are they? Hypotonic solution. First one is hypotonic solution. Hypotonic solution means low osmotic concentration. Okay, it is having low osmotic concentration. Why it is having low osmotic concentration? Because here solvent concentration will be solvent more, solute less. In which one? Hypotonic concentration. Okay, it's having very low concentration. Okay, that means per unit volume of solution, the solute dissolved will be very less. Okay, so here low osmotic concentration when we are comparing to the other solution, and when we are placing a cell in hypotonic solution, here we said low osmotic concentration. What do you mean by low osmotic concentration? Solvent will be more, solute will be less. Okay, so solvent will be more and the solute concentration will be less in which one? In a hypotonic solution. Okay, so here first about osmosis. This is very important part. You may get several questions. So here you may get what is osmosis? That is important question. And here we said semi-permeable membrane. That is important point. And here another one, the solvent movement. That is due to the difference in water potential. Okay, that is one important question, previous year question. Another one, osmotic concentration. That is based on osmotic concentration, there are three types of solution. This is also an important question. And which are the three types of solutions? I will tell: hypotonic solution, hypertonic solution, and isotonic solution. So first we are learning about hypotonic solution. So hypotonic solution means, this is also previous year question. What is hypotonic solution? Don't get confused. Hypotonic, hypo means less concentrated solution. So here solute concentration will be less, solvent concentration will be more. Okay, and if you are placing a cell in a hypotonic solution, what will happen? Generally, if you are placing cell means the cell sap will be there, or the cell inside the cell organelles will be there, and all the cell contents will be there. So inside the cell the concentration will be more, right? Cytoplasm will be there and the cell organelles will be there. So overall we can say the concentration compared to the hypotonic solution, cell will be having more concentration. That's inside the cell concentration will be more. So if we are placing a cell in a hypotonic solution where the solute concentration is less, see this is having hypotonic solution and we are placing a cell here. Okay, so this is a cell, or if you are placing RBC, red blood cells, what will happen to that? Compared to the hypotonic means solute concentration is less. Compared to this hypotonic solution, inside the cell, solute concentration is more and the solvent concentration will be less. So if we are placing two cells, what will happen? Passive transport will start, right? That means which is not required any energy. So until the concentration becomes equal from the less higher concentration area of solvent, okay, where the solvent concentration is more and where the salt concentration is less, from that the solvent molecule will move to that other area, right? Inside the cell concentration is more and outside it is less. Solvent concentration is more, solute concentration is less, right? So from here water molecule or the solvent molecule, here we can say the water, that will move to the cell, right? Because there the concentration is high. So to make it in an equal way, from here water will move to the cell and what will happen? That a cell placed in that in a hypotonic solution that gains water. So the cell will become in a turgid condition, or that swelling of that cell, it swells up or that will become turgid, right? So we can say endosmosis, here endosmosis. Okay, so here this question, this is important previous year question. Many question paper you will see the same type of questions. If an RBC is placed in a hypertonic solution or hypotonic solution or a isotonic solution, so you should know what is hypotonic solution or hypertonic solution or isotonic solution. Okay, and if it is keeping in that kind of solution, what is happening? From inside cell, hypotonic means solute concentration is less, but in a cell solute concentration will be more inside the cytoplasm, right? So from outside more water will move inside the cell to make the concentration less, right? So until the concentrations become equal or it is trying to reduce the concentration, water molecules will move inside the cell. So if water is moving inside the cell, what will happen? Cell will become turgid or it will swell up, then that is called as endosmosis. Okay, endo, to inside the cell osmosis is happening, right? Then next comes the hypertonic solution. Okay, this hypertonic solution, this is also a very important previous year question. Hypertonic solution, that means here high osmotic concentration is there. Why osmotic concentration is high? Because here solvent is less, solute more. It's a highly concentrated solution. And if we are placing a cell inside this highly concentrated cell, what will happen? Compared to this hypertonic solution, this is hypertonic. That means highly concentrated solution. Here you are keeping a cell. But compared to the cell, here concentration is more. Inside the cell, concentration is, sorry, inside the solution, this hypertonic solution, concentration is more. So what will happen? From the cell, the solvent or the water molecule will move to the hypertonic solution because here the higher concentration is there, right? So what will happen? The cell will become flaccid cell. Okay, that means cell loses water. The cell loses water. Okay, or exosmosis. Water movement is outside. From inside the cell, water will move towards outside. Okay, so these are the three answers you can expect. Definitely hypertonic solution or any type of cell which is placed in isotonic or hypotonic question you can expect. Okay, you should understand. Then isotonic solution means two solutions which are having the same concentration. Isotonic. Both the solutions will be having the same concentration. So there is no water movement, and if a cell is placed in an isotonic solution, that means there is no change in the concentration. Same cell concentration also will be the same. Solution concentration also will be the same. So there is no movement inside and outside. Okay, no water movement. Here, osmosis that helps in maintaining the cellular turgidity. Okay, osmosis. Then water transport, movement of water molecule is happening there, right? And that helps to maintain the cellular turgidity. Osmosis will happen until the cell turgidity is getting maintained. Clear? So here in the passive transport, we have learned about osmosis, facilitated diffusion, then diffusion. Next, what we need to learn is another type of passive transport, that is plasmolysis. Plasmolysis. Plasmolysis means shrinkage of protoplasm. Okay, shrinkage of protoplasm. We will say plasmolysed cell. If the protoplasm is shrinked and it is moving away from the cell wall, away from cell wall, protoplasm will get shrinked and it will go away from the cell wall due to exosmosis. Exosmosis, that means water is moving from the cell to outside. Okay, this is happening when the cell is placed in a hypertonic solution, right? Exosmosis we mentioned there. In a hypertonic solution, we are placing a cell, means inside the cell, compared to the hypertonic solution, inside the cell concentration will be less. That means solvent concentration will be more. So water concentration is more inside the cell. So water will move from the cell to the hypertonic solution. So what will happen? Plasmolysis will happen. Exosmosis is happening there. Plasmolysis will happen. So the water content will reduce so that protoplasm will move away from the cell wall. And when and all this plasmolysis will take place, this takes place in 3 stages. Plasmolysis in 3 stages. Which are the 3 stages? That means limiting plasmolysis, incipient plasmolysis, and evident plasmolysis. Okay, and the reversal, another important question. What is deplasmolysis? Deplasmolysis means the reversal of plasmolysis. Reversal of plasmolysis. That is called as deplasmolysis. Okay, so here plasmolysis we have learned, and next is imbibition. Imbibition. What is that? Imbibition, see, is a process of adsorption of water. Okay, adsorption of water by solid particle. Adsorbs water. That is called as imbibition. And there is no solution formation. They won't no solution formation. They will absorb the water. Which one? The solid particles of a substance that will absorb water, but there is no formation of a solution. So imbibition, that causes release of energy and development of high pressure. That means, see, wooden doors, if they are absorbing water due to during rainy season, if they are absorbing water, what will happen? Swelling of wooden doors will happen, right? So it may create a pressure. This absorbed water will create a high pressure. That's why swelling is happening, right? And that causes release of energy also. Okay. Then seed germination. While seed germination, what is happening? Water is going inside. Then it is exerting a pressure, right? That's how seed is germinating, and other vital processes are taking place, then the seed is getting germinated, right? So that's how imbibition is happening. So we have learned about passive transport. Active transport we have learned. Now passive transport, under that diffusion pressure we learned. Facilitated diffusion, in that about carrier proteins and channel proteins we have learned. Then osmosis. In osmosis we have learned about what is osmosis, then hypertonic solution and isotonic solution, then plasmolysis, then imbibition. Okay, this is about the means of transport. And in the next class, what we will learn about plant water relations. Okay, plant water relation in that absorption of minerals, then ascent of sap, transpiration, guttation, and phloem transport. About these concepts we will learn. Okay, so we'll see in the next class. Thank you.